Hello, thanks. So I'm Brian Haun. I woke up a little under the weather yesterday and it's persisting today, so sorry if I'm a little coarse. Let me try to share this presentation and we can get started. Can everybody hear me okay?
Yeah.
Right.
Coming through loud and clear, Brian. Thanks.
Yeah.
Okay, so hopefully this is full screen for everybody.
Looks great.
Great. So I probably have too many slides here, so I'll move quickly and try to save time for questions at the end. And hopefully I can stay within the 15 minutes that were allotted. I wanted to do something a little different and maybe have some fun with this talk and just kind of give this underlying tone of alchemy and transforming students into scientists. I used imagery generated by DALL-E, which is an AI art generator. And that's just to kind of also dovetail with this concept of innovation and trying to educate it with new technology. And you'll see if it's an AI image down at the bottom right here. I asked DALL-E 3 to make a logo for itself so that I could cite my source. And this was the logo it wanted me to use to cite DALL-E 3. So when you see that, you'll know it's an AI image. And as mentioned, the collaborators in the presentation here are Bulseco and John Berestecky. And my email, LinkedIn, ResearchGate are down here. Feel free to email me or connect with me if you have questions, thoughts, comments, concerns. All right, thanks everyone. So as an overview, we'll discuss our facility and the student research group that we have there, the education program that we've been working on at this facility, the research projects that stem from the education, and we'll finish out with some conclusions. So to give a quick overview, a little history of the facility at Kapiolani Community College, John Berestecky here on the top left developed this monoclonal antibody service facility and training center. Its acronym is Massive TC. And this was essentially a core facility that enabled the development of antibodies through hybridoma technology for researchers that had needs for antibodies that weren't commercially available. And this sort of grew into a larger education program that John Berestecky put together, where he developed courses for microbiology, immunology, cell and molecular biology, along with labs. And some of this was in collaboration with some folks that were brought into the fold, Matt Tudhill and Alan Garcia. And I want to highlight that these courses are largely 100-level courses. So freshman year at a community college, they're hitting the ground running with education in microbiology and immunology. And then there's a research experience that the students can do where they can sign up for 295, and that is basically research project credits at the community college level. And this was initially funded by a workforce development grant and has kind of emerged as a fully fledged program that mostly is now John Berestecky, Alan Garcia, and I, largely funded by Embry at the community colleges I mentioned, offering education and research programs. And the magnifying glass is to highlight that it's largely focused on antibodies and the technology of antibody development and engineering. And so some of the key components of the research and education are hybridomas, obviously, antibody sequencing, which can be quite challenging, antibody characterization, development, engineering, and so forth. And we now are able to support TAs through the Embry program that helps support the classes and the labs. As mentioned, we have a student research group that the students often can grow into through the education program. And this is extracurricular research. The acronym for this is CARM, standing for the Kapiolani Association of Research and Microbiology. It's a student research group where undergraduates get to learn biotech skills. And through our mentorship, they conduct comprehensive research and get to present their findings every year in the form of a poster presentation at a local symposium at the medical school here, which is always a very exciting time in April. And we are now supporting postbacks. Usually, although it's a community college level, our students typically stay with us as they go on to the main University of Hawaii campus and continue their research all the way through postbacks, and often even into grad school, still maintain relationships with us. And so we can move from there into an overview of the education program. Well, let me first say, are there any questions about this setup that we have going on here? Feel free to conversate. All right. Then it's freezing up on me. Okay, so then let's move into the education program. And so this is what we sort of came up with and how to take molecular cell biology basic core courses and try to blend that with really educating students on antibody technology education, which we sort of thought that we could educate genetics, proteomics, and cell biology all through the lens of antibody development and immunology. So we have two courses here, Micro 230 and 240. This one is in the spring, this one in the fall. And they're both cross-listed as 275 and 275L, which gives them credit for the 275 credit needed at the main campus at Manoa when they transfer for their full bachelor's. And so we focus on DNA technology in the spring course, where the students can learn how to perform a number of sort of basic molecular assays from RNA and DNA extractions and manipulations, recombinant expression of DNA, do some computational analysis, and develop some novel constructs in the form of expression vectors. And then the idea here is that some of the information that is generated through this feeds the research group, which is outside of this circle. The research group then can feed into the coursework, and we get sort of this nice collaborative environment. And then what the students develop in 230 gets transferred into 240, where the vectors they develop here are now expressed, and a little more is dug into with the proteomics side. And so protein purification and assays and more computational design. And we can learn to use certain tools to assess how good these antibodies might be. And that can go into the next course to develop maybe a better iteration of that expression vector. And so that's the overall concept here. We're still working out some of the kinks, but it's been a really exciting journey as we sort of just get started piloting this concept. And so under this design, test, build model, and in collaboration with Dylan, Sandra, and Todd, we were able to put together an NSF proposal, largely spearheaded by Dylan. Great efforts there. And we were awarded this advanced technical training grant. And so that's been fantastic. And I guess part of why we're able to share this with you all here today. So I think I sort of went through this in the last slide. But just to zero in on 230 a little more, this is the spring course. Some of the outcomes they will learn as far as technical skills would include pipetting exercises and expertise, DNA extraction, PCR, RNA extraction, reverse transcription PCR. We actually do Gibson assembly while we do educate on some more traditional cloning techniques. They will perform and analyze Sanger sequencing, which is always a very exciting concept at that level, sequence analysis. And we use a Chinese hamster ovarian cell expression platform. So they get to learn about that. And we do multiplex luminescence assays to assess antibody performance, or whether or not they're actually binding and making constructs that work. And then 240 will go further with the proteomics side, still getting expertise in pipetting, which is obviously fundamental. This is exciting as they get to kind of get the concept of how dirty our environments are through bacterial cultures of the environment. Through that, they learn microscopy, and then they kind of get a really good sense for aseptic technique. And then they move into eukaryotic cell culture and they get to learn really how to do that. Of course, we incorporate PCR, Sanger sequencing, expression, some flow cytometry, and Luminex assays as well. Finally, their evaluations are largely in the form of either weekly quizzes or some in 240, they will do actual book report on a research article. In the 230 course, they'll do a PowerPoint presentation on a research article, which is sort of what this is highlighting. In both classes, they have to make a poster presentation for our local undergraduate research fair at the Kapiolani Community College. And so through these exercises, they're also learning how to discuss their scientific findings or interests with the scientific community. And so we're also trying to build the communication skills, which I think the pandemic really highlighted that awkward science folks like ourselves probably need to improve in that regard. And so are there any questions about the education program?
Hey, Brien, we're gonna hold the questions until the end of both the talks.
Okay.
Thanks.
I don't get to see how good I'm doing on time. I feel like I'm running out, so I'll try to blaze through research projects and conclude this. So I'm trying to highlight an overview of some of the things that the research group works on. That would be CARM. And so we do these multiplexed assays through Luminex technology. Largely, our antibody efforts have been focused on viruses such as SARS-CoV-2 and Marburg, and we'll attach the spike or glycoproteins to these beads, and then we can incubate with whatever our primary antibody source is, whether that's serum or purified monoclonal antibodies. We wash that out and we detect it with a PE label detection antibody and then run it on the MagPix machine. And this is highlighting one of our recent Marburg virus antibodies that recognizes the glycoprotein. And we were able to do some epitope mapping for it looks like it binds a region that has not yet been crystallized. So we had to make this theoretical green line here of where the epitopes actually are through our collaborators Integral Molecular. And we also have single cell RNA-seq libraries from Zika virus that we're sort of moving through and doing this type of analysis with as well. And if I didn't mention, that would be Zika virus. We are teaching the students CRISPR-Cas9 techniques where we try to edit certain antibody genes in different B cells and perform different flow cytometry and other sort of molecular assays such as quantitative PCR to look at how well we did in editing the genes. And a large focus of ours is on VDJ sequencing, which is a really complicated set of genes that come together to form the antibody binding domains. And because they're so variable, it can be really difficult to sequence these properly, especially in hybridoma settings. And so we use different primer techniques, assortment of primer techniques, template switch oligo techniques that the students develop to really try to zero in on efficient ways to sequence these things. And through that, we combine that with some of our B-cell enrichment assays we're working on that can help us analyze antigen-specific B cells, combine that with our VDJ sequencing techniques, and use our newest toy, a nanopore MinION, to do some next-generation sequencing. And our undergraduates get to learn how to do this and how to analyze next-gen sequencing results through Python and Linux command line. And we're still working on developing this, but it's been a very exciting time in that regard. So we also do some engineering where this is supposed to help us try to reduce the cost of recombinant expression and large library screening, where we take a traditional monoclonal antibody, which would usually have a light chain and heavy chain variable domain on two separate vectors or as two different genes. And we link them together as a single chain variable domain conjugated to a human IgG1 FC for detectability and easy purification. And this has worked out really well for us and basically reduced the time and cost in half for us to screen these large libraries. And working on developing interesting assays to detect inhibition of viruses, in this case SARS-CoV-2, from their cognate receptor, in this case ACE2, and learning how to use this technique to screen our hybridoma supernatant for antibodies of interest to carry through further development. In this case, highlighting several clones that have inhibition to different strains of SARS-CoV-2 at a single dilution point, which was very exciting for us to figure out. We have now a surface plasmon resonance machine, so students are getting to learn how to use this to actually determine discrete binding values for affinity, association and dissociation constants. And so this has also been a really exciting avenue for us to move into. And some of our antibodies actually have very high affinity constants where we would not have otherwise been able to determine that through our Luminex assay. And computationally is exciting things that we're moving into here. In this case, I'm highlighting our use of the SobPred toolbox, where we can take our sequences and see how valuable they might be in the therapeutic space. And we developed a way to take their histograms and raw data scores, develop a metric through that, whether they're bad, medium, or good. And then we could actually run principal component analysis and see what of the 5 components listed here are giving these good values or bad values for therapeutic developability. And the idea here is that we can then modify those sequences to develop a better and better antibody, which would feed back into the courses, as mentioned in the education earlier. And so that concludes the research projects. And this will be our final slide here. So at Massive TC, we're cultivating a crucible of workforce excellence through the alchemic blend of education and research, transforming foundational concepts into technical prowess, equipping students with the tools of modern science in the education space. In the research space, our students distill technical expertise, crystallizing into the next vanguard of biotech leaders. For innovation, we're harnessing the philosopher's stone of education, transmuting undergraduate students into scientists. And the alchemy of it all, as an overview, would be through our collective quest. We transfigure the scientific community of Hawaii, precipitating a nexus of innovation and mastery. And so I'd like to thank everyone, our undergraduate students, our leaders at Massive TC, and our TA who works diligently to help us, and of course our collaborators with these different things. And with that, I thank you, and I'll yield for questions at the end.
Thank you for your presentation on how you're guiding your students. At this time, please join me in welcoming our second talk speaker, Dr. Chandra Arora. Dr. Arora's presentation is titled Galvanizing Gaps in Classroom to Career, where in addition to resumes and mock interviews for college to career readiness, project-based learning proved to be tremendously useful. And as a reminder, at the end of Dr. Arora's presentation, we'll open it up for a Q&A for both of our Project Talk presenters. Thank you. Dr. Arora?
I see a note from Dr. Aurora in the chat saying that there are internet challenges.
Oh, yes. Let's see.
Maybe we should have a little bit of time for questions now while she's working some of those out. What do you think, Dion?
Oh, let's see. Dr. Aurora cannot unmute. Cassandra, can you unmute?
No, I can just ask them to unmute. Maybe we'll switch to Q&A, and if you can log out and then log back in, maybe.
Okay. Did you hear that, Dr. Aurora?
Or are you sure you can't unmute her?
I'm sure
Yeah.
I have
Oh.
the power. I have the power to mute, and I have the power to ask to unmute, but I cannot unmute.
Oh,
Mhm.
okay.
Yes, if you can just. Okay, so that's a great suggestion, Sandy. If we can go ahead and just move to Q&A for our first talk. I can open it up. You can use your raise your hand function.
Sure.
Can I ask a question without raising my hand? Brien, I was wondering,
Yeah.
how many students do you have in your course?
The course tends to fluctuate. I think right now we have 7 enrolled. It's not very high, which is wonderful. So they get a lot more hands-on experience. I think around 10, up and down, maybe 3 is probably the average. As we get this ironed out, we'll probably look for ways to reach out and grow that if it's feasible. But it's been exciting to figure out how to, accomplish these things. And I'm glad that it's not large classes like it could be at the main campus, because I think this would be very difficult to accomplish if it was. Good question.
No,
Yeah.
I can see that.
I'll add to that, Sandy. Yeah, so the 230 and the 240 classes have typically been running somewhere between 5 to 9 students for the last couple of semesters. And the other thing about them is that we're in the process of retooling them. They used to be just general tissue culture and molecular biology, but now with Brian's addition and Brian's help, they're turning into this antibody engineering kind of paradigm and focus. The immunology class, which is the micro 161, is actually burgeoning at the seams. We're now offering two sections of that, and part of the reason for that is that it's also a service course for our medical lab technician program, and we're actually trying to get a lot of those students into our program also, especially the really good ones and the real interested ones. And then our general micro classes, we offer multiple sections. So I would say we probably have something like about 60 to 80 students total in the general micro classes, the 130 and 140 classes.
No, that's sounding great. And Beth had a question about oh, hey, Chander.
I'm trying to connect with my phone and it's echoing now.
Hey, Chandra, it's Russ. I think you're connected twice.
You
Yeah.
are. Let me kick your other device out and it'll stop the echo for you. Okay, that should hopefully fix your issues.
Are you able to share your presentation, Dr. Aurora?
And this is on my phone. I only have it on my computer, the presentation. That's a problem I'm facing. Computer is not letting me unmute at all.
Okay. Do you want to send your presentation to me? You can email it to me.
Okay, let me do that right now.
And we can continue with the Q&A while we get that sorted out in the back end. Thanks for your patience, everyone.
In the chat, Elizabeth Stewart asked a good question about if we were querying local industry for what skills might be of use in your courses, or do a more broad survey of needs to decide on skills. And that's something we recently been talking about, and it's a really great point. I think there were some folks at the NSF ATE meeting that I wanted to reach out to that were doing that type of work, specifically for immunotherapeutics. We haven't got around to it yet, but it's definitely something we're thinking about. So that's a great question / suggestion.
And I might add one thing to that as well. One of the local biotech companies in Hawaii is actually on our advisory board, and I'm working to meet with them now to look at the the workforce needs and what the gaps might be and what they're looking for. But I'm also actually meeting with the Mass Bay Community College on Monday, and they actually have one of the director there is actually also on our advisory board, and I'm going to take a lot of the feedback that they're providing, because they've done an amazing job at developing their biotech program there, and see what we can consider and look at it at how we might integrate that in the future in Hawaii as well.
Yes.
I was wondering, where do your students go geography-wise, generally? Have you been able to track to see, do they stay local or head to California or?
Yeah. A lot of times they sort of grew up here and have family here and want to stay local. And you might imagine that tourism is really the focus of Hawaii. So that's something we've been battling with and discussing a lot over the years, is how can we build a biotech type of environment here where students would have the opportunity to stay here. So they'll either work for the Department of Health, go on to graduate schools here or on the mainland. And some folks have ended up in biotech programs on the mainland. There were some that were actually running the SARS-CoV-2 testing facilities here at the airport. And so our students do end up going to great places, including med school. John might be able to speak to more of the history of that than I could, but typically if there's a position or graduate school, med school for them here, they'll usually do that. Otherwise, if they usually go on to a biotech or med school on the mainland.
Yeah, I would confirm that. We haven't really tried to formally track our students, so a lot of this is just informal. We hear back from them, but most of our students are on a 4-year track. They'll start off at the community college. Many of them don't know exactly what they want to do, but we kind of help focus them, and that ends up sending most of them on a trajectory where they're going to complete a bachelor's degree. And there aren't really that many opportunities here in Hawaii in biotechnology, very, very few. I would say probably the most opportunities are in research labs throughout the University of Hawaii system, and a couple of the other universities here, Chaminade University and Hawaii Pacific, also have graduate programs. So many of them go there. And then, the students I've heard back from are all over the states. A number of them go off to Seattle. There's quite a hub in biotechnology there. Several have gone to the East Coast. Several have gone to San Diego, San Francisco area. Yeah, but again, most are end up going into graduate school, going on, finishing a 4-year degree and going on to graduate school that I've heard of. Yeah, so all over the place.
And
The
this is,
skills? Oh, go ahead.
well, I was just going to add, and this is actually something I'm working with the Hawaii Public School Foundation as well in developing this biotech locally in Hawaii, because that really is something that is lacking, and we're looking to find ways to diversify the economy there. And I grew up in Hawaii, and I left 40 years ago because there were no biotech jobs for me. So I've been on the East Coast now for 28 years. And I see the need in Hawaii. And so that's the reason I'm working with several institutions in Hawaii, promoting STEM and STEM ed and looking at ways to grow it there.
Well, certainly if they come to the mainland, I mean, we can see it on Biotech Careers. There's at least 450 companies that list antibodies as one of their chief business occupations.
Absolutely.
So the skills they have and the skills you're teaching them, Brien, will be really, if they're willing to, leave Hawaii, the skills they have will serve them well.
Yeah, thank you. That's a good point. As antibody therapeutics continue to kind of grow and become more and more sort of important for broader ranges of disease, we kind of see that as being a little niche education field that could use a little bit more. There's not really a lot of courses you can take where you're really going to learn the nuances and details of this and how to train in it. But alongside of that, it's such complicated genetics and things like that, that they learn skills that are translatable into any other type, for the most part, of molecular biology that they might want to get into. So oftentimes they might not stick with antibodies anymore, but when they go into their graduate programs, they have learned so many fundamental skills and so many complex assay design and development that they really take off and become sort of key students, graduate students, or key members of wherever they end up. So it's kind of been exciting to see that happen.
No, that's great. Dion, how are we doing with the slides?
I have not heard back from Chandra yet. I can see that she may have been trying to log back into her computer. And so, Chandra, would you like to reschedule? We can do that, or we can just continue on with any more additional questions we have for our first project talk presenters, given the fact that it's about 18 minutes the hour.
Well,
And so did we have any additional questions for our presenters? I found it interesting, Brien, that you mentioned the pandemic and getting your students to present. And do you feel that your own hesitation, because you also did mention that you need to work on that yourself, do you think that that sometimes has a direct bearing on your students?
Yeah. Yeah. So I don't know, that could be a personal feeling of mine, but I think that the public isn't used to seeing science live and direct, and how things change as we learn more, and it's discomforting to them. And it felt to me like this was should have been a learning lesson for science communication. And obviously science education in the US isn't necessarily the best. So there's already this room for a lot of, I think, frustrating theories to develop among the population. And I found that when discussing with almost anybody, anything about the pandemic, the vaccines, it usually became absolutely exhausting. And I'm sure many of you have had that as well. And so initially our students are really terrified, and then they push through, they get better. We make them present a lot in our research group as well. We have our weekly meetings. They rotate who gives a PowerPoint presentation on their work. They're also very terrified in the beginning, but after, a few times they get better, and then they go on to Manoa, the main campus, and they'll tell me, they make us present a lot, and I feel so comfortable now. And it's
great.
not so scary. And I'm like, yeah, that's exactly, what we want to see. So it seems to be working. For my own personal experience, I don't know. It's kind of interesting. How do you communicate difficult things in ways that are easy to understand is something I'm always trying to work on. It's challenging, and maybe something for us all to kind of, work towards.
Yeah.
Thanks. Hopefully that answers questions there.
hey, on that note, I really want to encourage you to have your students prepare. If they're doing posters already, right? It's not that big of a step for them to start writing papers. And we have a journal with the ATE program called the Journal of Advanced Technological Education. And the version that will be published this summer and handed out at the conference in the fall, it is focused on undergraduate research. And so it'd be really great to have some of your students writing something about their experience.
Yeah.
That is a wonderful suggestion, and thank you for letting me know about that. That is one of the other main goals of mine with this program and our group. a lot of what we do, I think, could really stand up to external review and get published. One thing I found is that the writing communication is also terrifying to them, as it is even to me. Dealing with reviewers currently, sometimes it's just challenging. So that is something I have to learn how to get better and motivate them. I'll ask them to write up their methodologies or a background, and often they think that I forgot, and I can't squeeze that out of them yet, but I'm working on it. Thank you. Yeah.
And I'm on the editorial board. Feel free to reach out with any questions.
Awesome.
And Beth
Thank
put
you.
the link. Beth put the link in chat too. And you have another question here about doing lab skills testing with your students.
We did that initially. What we like to do is, once we teach them pipetting fundamentals, we have them do a dilution series that we measure so they can look at how well they all did in a dilution series to kind of assess their maybe weak points and how good that their pipetting skills are. We obviously don't really grade them on that, but we try to highlight, areas where they could improve, and that's about the extent of where we're at with that right now. Probably we could improve on that though. So that's a really good question. I think pipetting skills is really our focus on skill testing, and maybe it's the easiest to test as well. And we like to, if we have the time at the end, have them recreate those curves, and you can watch the standard deviation go from all over the place to everybody just being a perfect curve, which is really cool to see. And I think encouraging for them as well, that they can physically see their improvement. So thank you.
And with your Sanger sequencing, you can certainly get a lot of quality information.
As an illustration for the need in Hawaii, I'll relay a story I just heard about yesterday. During the pandemic, the Department of Health did not have the capacity to characterize the COVID variants that were showing up in Hawaii. And they actually turned to high school students. There's this INA network in Hawaii that works at the high school level that does sequencing and assemblies and characterization. The Department of Health actually turned to high school students to do real work in characterizing the variants that were showing up in Hawaii, because there was no bandwidth to actually do that at the Department of Health. So that's kind of, showing what the need is, and there are ways that, hopefully we can fill that. And so, I'm excited about the prospect of doing that, moving in that direction.
Yeah, there was
to add to that for a second, Brien had mentioned it during the COVID epidemic. They also didn't have the capacity to do all the testing that needed to be done.
That's right. And
so they reached out to us and asked if we had people that could help with that. And in fact, we did. We had two people, as Brien mentioned, that ended up being very pivotal point people on the testing facilities that emerged here. And in fact, they ended up supervising med techs and others that were working in the lab. So yeah.
Wow.
Chander's ready. And I know that we're short on time, so during her presentation, if you can please put any questions that you may have in the chat, and we can share her answers, and we can share that with you on the website. With that, please join me in welcoming Dr. Aurora.
Thank you so much for your patience, everybody. I truly apologize. It has never happened to me. But anyway, without any further ado, I'll try to be as fast as possible. I really apologize for that. I'll take the total blame. You want to reschedule, I'll be very happy to do that. But I'm using my phone audio, so if it is not clear, again, it is the technicality. This is what, nothing high-tech, nothing like what you just heard, a very high-tech presentation of antibody. This is anti-employment and anti-unemployment and I would say, equity that I'm going to talk about. Very basic thing, how I engage students in all these things. So we are located in California, in Silmar, exactly along the West Coast. And this is LA Mission College, and we have a stackable certificate program. The first certificate is done in one semester, and this has no prerequisites. People who are new to science but are interested in science are mostly the students we target. And each certificate is stackable. After first certificate, most of the students, they get employed, they come back, continue their education, get the second certificate, get the promotions, get the third certificate. And we are proud to say we are one of the first LACCD colleges that we got bachelor's degree approved just recently for biomanufacturing. So we target all these populations, and I have introduced a different pedagogy for the students, which is contextualizing soft skills that I want to talk about. It's called project-based learning. So these were the gaps that we recognized. Number one was equity and engagement. Everybody came with a different background. It was not equal opportunity. It is bringing the best out of everyone with their special potential and giving them what they need to be their best. And how we do that by recognizing what their challenges are and how much each of them needs to overcome those challenges. So leading by example, setting some rules right from the beginning, from the welcoming letter itself, creating an environment, and nothing creates a better environment for diversity than over a candid breakfast, which was even during pandemic, maintaining safe distance. At the bottom, you can see just starting with what we are doing and how we discuss everything in class, making those rules very valid. Second challenge or second gap we realized was employment. And I thought hands-on training and plus soft skills, if we tell them about them, this will get them employment. And this was a little bit modified with the pedagogy I want to talk about. So how we galvanize these gaps by project-based learning, starting with scientific method, making them understand what these project phases would be, driving a question that comes from their mind, and getting an answer in the end to that driving question or the objective. So starting with a driving question, in the end, they have to come up with I build the team, help them have some results, make a roadmap, and in the end, they have to answer that driving question. So this is what we do in the inclusive classroom, making a project-based learning, starting from the beginning, where they bring the projects to the class, what they want to do. Starting with a daily problem that they can they can relate to, and making their team with a diverse group where they have assigned roles. One is operator, one is verifier, one is note taker, and one is a team leader, and they rotate that. Believing in student strength and really creating their best of their potential through their minds. We do something like an engagement of every student where they can write their ideas about the project and how they will put it together as a main question. So under the guidance, they will start doing that. Team engagement starts from day one, team discussion, and then notes taking, which they will share with the whole class. So doing that, our attendance and student retention really emerged, very much improved. And end of this, we do the biotech exhibition where they have to present what was their idea they wanted to do and what is the result. So they make a roadmap, which is a flow chart right here in the middle before they do anything. And everything is discussed openly in the class by the team and the team leader. Then they conduct the experiments, the problems they face, the troubleshooting, the root cause analysis. All of them they learn in those 8 weeks. 8 weeks they learn the techniques, 8 weeks they apply to a project. And this is the end project from the student teams that came up: bioremediation of edible oil waste using bacteria, sugar preferences and their oral microbiome, nurturing gut microbiome with turmeric. Somebody came up with that idea. So it's all what they relate to and what they come up with. So bioinvaders in swimming pools, they thought of doing that. And this is where they present, and all the employers and people are invited. It is open to public. They really gain a lot out of this. So these are all students presenting their projects. These are the skills they validate while they are doing the projects. These are, at least they have to use at least 2 of these techniques in doing a project. Even if they want to bring moon and stars to the lab, they have to employ the techniques. And what gets contextualized here is the soft skills. They learn how to do the teamwork, time management, root cause analysis, problem solving, the best of all, sense of engagement and pride. So I used to think this is the model that will work for employment. And with project-based learning, I thought this is what we needed to do. We need to change something here. And that's what happened. Now that plus sign, hands-on training plus soft skills became the multiplication sign. So project-based learning strategically gives hands-on training, and soft skills are automatically included in that. It's contextualized in that. And this is what equals to employment. The difference being, if one of them is 0, the product will be 0. That's the difference between plus and the multiplication. So hands-on training and soft skills together now in this model result in the employment. And when it came to inclusion and engagement, this was critical thinking, teamwork, motivation, everything increased. This is student survey for 2.5 years, and we published it in Journal of ATE. And the success definitely owing to our industry partners. Our industry partners come to our campus just to interview our students, the biotech students only, not for a job fair. This is Takeda team. That was Grifols team. They come to our campus. And these are some of the students that got hired after 1 semester of training and contextualizing these soft skills. This guy on the right, he had a PhD in food technology, still came through this. Just recently, 2 students, they were hired and they were so excited, sent me their badge right away. Another student who had never seen a science lab right in the middle, he's working as an intern with stem cell research now. So this was our employment, and this was our technical success in every semester, pretty much 100% employment of these students. Some of them had never done the work that they are now doing as a team. These are all the students. These are the employers that I work with, and I'm very pleased to say they literally wait for our students, and these students are continuing education, and now 25 of them are enrolled in bachelor's program, which is starting in fall semester here. So some recent publications that we did, some panels that I was invited to present this model and to actually explain how this is being done from classroom to career so successfully. I was very happy. Nothing like what Russ Reed just recently shared. I'm just going to share a stage, something smaller with the senator. I was invited to talk about the success of this. Celeste Carter mentioned it, and I was very honored that she actually remembered this is our employment rate because of this contextualized training. These 2 students were invited for a TED Talk because of how they succeeded in their career. So some of the comments, and one of the congressmen, he gave us $400,000 to buy new equipment when he came to know this is what we are doing. These are some of the publications. I'm just zipping through, I apologize. And it was also in connections that it was published, the same model. So what we are partnering now to get in these people interested who are in between or affected by pandemic, working with America's Job Center. So this was getting a lot of attention and funding from our chancellor and from rest of the people in our area. So this is what I presented in Meeting of the Minds for CWA, what we are doing for development of workforce recently. And this is our first publication on contextualization of soft skills that I was mentioning, Journal of ATE. And another one recently came I'll skip these. It's not about me. I got some, they didn't know what to do with me, so gave me some awards. Now we have the bachelor's degree and we are diligently working on that. This is a cell and gene therapy certificate coming up in summertime that is fully funded. And creating this, we got some partners, some workforce centers, and developed with employers a biotech credential certification and Amgen Biotech experience to tap into. This is with Grifols and Takeda and 7 other industries that we developed this. So developing the high school program, every summer I hold a high school workshop and they get a certificate in the end of it just to expose them to biotechnology. Otherwise, it's a very intimidating word. So we held in our lab, the Amgen Biotech Experience, the international meeting, which was a great success, and people from 17 countries, they actually participated in that. So creating the BioTC certification is hosted by Biocom, and our Animation College is the hub for that. We got in the process $10 million to make a training center based on our model from our assemblywoman, and we are building a new institute. And when this was publicized, our congressman gave us another $2 million to get some equipment, which we are in the progress of getting. I think most precious gift today is definitely your time and attention, and I'll stop it here. I really apologize once again to everybody.
We all encounter rough moments with technology. Thanks for your perseverance. It's quite a tour de force.
I have never felt so embarrassed as I felt today because I still cannot put my video on or the thing on my computer. Whatever is the reason, I think it's to torture me and all of us.
And we will invite you to come back if you would to have a little bit more time, but we thank all of our presenters today, and Cassandra put a link to the survey. Please feel free to click on that and provide us with feedback. Again, thank you to our presenters, and we will be posting this on our innovatebio.org website by Monday afternoon. And again, we understand technical difficulties. It happens. So
thank you.
Everyone coming. And no worries at all. It's Friday, and we wish you all the very best weekend. Stay safe.
Thank you. Thank you, Dion. Thanks, Russ, for being here and encouraging. Thank you. Thanks, Sandra.
Excellent. Come back. Love it.
Thank you. I will.
We want to hear more about that cell and gene therapy certificate.
Thank you. Thank you. Thank you. I'll definitely reschedule it. Have a wonderful one, all of you. Thanks again for your patience.
You had a lot of great information, and
yes, that's right. Yeah.
Yeah.